Hello everyone, welcome back to NEET Master Series, and you are watching the second part of our molecular basis of inheritance MCQ practice series. In the last video, we tackled some challenging questions to sharpen your understanding, and today we are back with even more. Get ready to dive in deep into the concept, test your knowledge, and ace those NEET exams. Make sure you have your pen and paper ready because we are about to solve some crucial questions that can boost your preparation. So let's get started. Our question number 21 is: assume that you chemically label both strands within a molecule of DNA. You then allow this DNA to replicate using unlabeled nucleotide. Which of the following statement about the two resulting DNA molecule is false? Option A: both will have the chemical label. B: one will have the chemical label, the other will not. C: one strand within each molecule will have the chemical label. And D: assuming no replication error, both molecules will be genetically identical. So our answer will be C, that is one strand within each molecule will have their chemical label. Okay. So, each strand of the original molecule serve as a template for the formation of a new strand. Thus, each new molecule will have half of its nucleotide and the chemical label from the original molecule. Okay. So, our next question is, that is 22. The process of gene transcription begin with the option A: binding of RNA polymerase to a region of DNA called the promoter, removal of neutrons from the nucleus, C: joining rRNA with various hydrogen, and D is attachment with mRNA. So our answer will be A, that is binding of RNA polymerase to a region of option B, C, and D. These options the next question is option A. So polypeptide that regulate the expression of groups of genes during development. Okay. So our next question is, that is 24. During transcription, the DNA site at which RNA polymerase binds is called enhancer, promoter, regulator, receptor. Answer will be B, that is promoter. So regulator is a gene which forms a biochemical for suppressing the activity of operator gene. While promoter is the gene which provides the point of attachment to RNA polymerase required for transcription of structure genes. Our next question is 25, that is during translation initiation in prokaryotes, a GTP molecule is needed in A. Association of 50s subunit of ribosome with initiation complex, B. Formation of formal met tRNA, C. Binding of 30s subunit of ribosome with mRNA, and D is association of 30s mRNA with formal methionine tRNA. So answer will be D, that is association of 30S mRNA with formylmed tRNA. Its explanation is that for the formation of initiation complex during translation of mRNA, GTP is also required. Okay, the initiator is AUG, which codes for the formal methionine in prokaryote. So, our next question is 26. Which one of the following triplet codes is correctly matched with its specificity for an amino acid in protein synthesis or as a start or a stop codon? A. UAC tyrosine, UCG start, UUU stop, and UGU leucine. Answer will be A, that is A UAC tyrosine, because AUG is an initiating codon, right? Initiating codon. And UCG codes for serine, and triple U codes for phenylalanine, and UGU codes for cysteine. Okay, so our answer will be A, that is UAC tyrosine. Our next question is 27, that is protein synthesis occurs A on ribosome present in cytosol as well as in mitochondria, B only on ribosomes attached to the nuclear envelope and endoplasmic reticulum, C only on the ribosome present in cytosol, and D on ribosome present in the nucleolus as well as cytoplasm. Answer will be A, that is on ribosome present in cytosol as well as in mitochondria. Its explanation is that ribosomes are the site of protein synthesis. We all know that, है ना? Mitochondria, being a semi-autonomous organelle, has its own protein synthesizing machinery. Okay. So next question is 28. Which step of translation does not consume a high energy phosphate bond? A. Translocation, amino acid activation, peptidal transferase reaction, and D. Amino acid tRNA binding to activate ribosomal site. Answer will be D, that is amino acid tRNA binding to active ribosomal site. Its explanation is during the activation of amino acids in the presence of magnesium ion and ATP, an amino acid gets attached to a specific enzyme, that is amino acid tRNA synthetase, and pyrophosphate is released, which breaks up to release energy. During translocation in the presence of enzyme translocase and energy from GTP, the ribosome moves in such a way that the peptide bearing tRNA of A site comes to lie on the P site, exposing a new codon at A site. In the peptidal transferase reaction, energy is provided by GTP. So that is all about our question number 28. Now come to 29. That is degeneration of a genetic code is attributed to the A. Third member of a codon, first member of a codon, second member of a codon, and D is entire codon. Answer will be A. That is third member of a codon. Its explanation is according to the wobble hypothesis, tRNA anticodon has the ability to wobble at the 5' end by pairing with even non-complementary base of mRNA codon. It corresponds to third base degeneracy of the codons. Okay. Come to question number 30. That is, the following ratio is generally constant for a given species. Option A, B, C, D. Our option will be C is correct because G plus C divided by A plus D. According to Chargaff, furines and pyrimidines are in equal amounts. Furine, that means adenine, and is equimolar with pyrimidine, that is thymine, right? That is thymine. And purine, that is guanine, is equimolar with pyrimidine cytosine. Base ratio is specific for species. Okay. So come to question number 31, that is eukaryotic chromosome. A, circular, are circular and contain origin and terminator sequence, are linear and have origin and telomere. C, contain coding and non-coding sequence. D, both B and C. Our answer will be D, that is both B and C, that is prokaryotic chromosome are circular and contain termination sequences. Okay. Come to question number Come to question number 32, that is the ends of eukaryotic chromosomes are single-stranded and must be bound by transcription complexes, are highly repetitive sequence that hold replicated chromosomes together, shorten every time a cell divides. D, require telomeres to move to other site in the chromosome. Answer will be C, that is shorten every time a cell divides. because the ends of eukaryotic chromosome are bound by telomerase, that is not transcription factors, and centromeres are not telomeres, hold the newly replicated chromosome together. Transposons do not require telomerase to move to other side on chromosome. Come to next question, that is 33. Which of the following would you not expect to find in prokaryotic DNA? A. Million of base pairs, histone proteins around which the DNA is coiled, functionally related genes grouped together in the same section of DNA, a majority of DNA that codes for protein or RNA. Answer will be B, that is histone proteins around which the DNA is coiled, because histone proteins are used to coil the large amount of DNA found in eukaryotes so that it can be packaged more efficiently within the cell. Prokaryotes do not face this problem because they have much less DNA. Next question, that is 33, the 34. The primary purpose of Griffith experiments on the Streptococcus pneumoniae bacterium was to A. Find a cure for pneumonia in humans, prevent cancers caused by exposure to UV, and determine if DNA is a hereditary material. D. Discover the molecular structure of DNA. Answer will be A, that is to find a cure for pneumonia in humans, because Griffith was a physician trying to find out a way to effectively treat or cure pneumonia. Nothing much was known about the structure or function of DNA at the time he did his work. Okay, next question. Which of the following part of DNA molecule are held together by hydrogen bonds? The carbon within the sugar phosphate group, the carbon within the nitrogen containing bases, nucleotide bases on opposite strands of the helix, and D. Successive nucleotides within a single strand of the helix. Answer will be C, that is nucleotide bases on opposite strands of the helix. Okay, because hydrogen bonds connect the bases on opposite strands of the DNA molecule, which is the reason that the helix can be unwound and separated relatively easily. All other bonds in pile molecule are covalent. Okay. Next question, that is 36. The primary reason DNA was first thought to be poor candidate for the hereditary material was that A. Griffith experiment showed that protein, not DNA, caused transformation. B. Viruses lack DNA yet still pass genetic information between generations. D. C. That is DNA was believed to have a simple chemical structure with little variability. And D. The work of Hershey and Chase showed that protein was the genetic material. Answer will be C, that is, DNA was believed to have a simple chemical structure with little variability. Because since the building blocks of DNA were thought to be structurally simple and few number, biologists prior to the 1940s thought that only proteins could have the necessary complexity and diversity to the hereditary material. Next question is 37. Consider Griffith's experiment on transformation in Streptococcus pneumonia. Now imagine that you are extending these experiments by injecting a mixture of heat kill strain R bacteria and live strain S bacteria into the mouse. The result will be that the mouse will find live strain bacteria in its blood and you will die, that is R, live R, die S, and live S. Answer will be C, that is diet. Since strain R, the bacteria, do not harm the mouse, this experiment is the equivalent of injecting the virulent strain S only. Thus, the mouse will die and you will find living strain S bacteria in its blood. Now, come to question number 38. A short sequence of bases on one strand of DNA is this. If this sequence serves as a template for the formation of new strand of DNA, what will be the corresponding base sequence in the new strand? Answer will be B is this. The complementary base pair will pair with each base on the template to form the specific sequence shown. Okay. All right. Now we have come to the final two questions of this session, but this time I want you to take these as a challenge and solve them on your own. I will display the question on the screen, that is 39 and 40. And I encouraged you to pause the video, try to answer them, and then share your answer in the comment section below. Let's see how well you have grasped the concept, and I'll be looking forward to see your answer. Don't forget, practice these questions will make all the difference in your NEET preparation. So here are your challenge questions. Okay, that's it for today's session. I hope you find these MCQs helpful in strengthening your concept on molecular basis of inheritance. If you enjoyed this video, don't forget to give it a thumbs up and share it with your fellow NEET aspirants. Make sure to subscribe to NEET Master Series and hit the notification bell so you never miss out on our upcoming practice session. If you have any doubt or question, feel free to drop them in the comment below and I'll be happy to help you out. Until next time, keep studying hard, stay focused, and remember your hard work will pay off. Happy studying. Bye bye.